So, hello, good morning to everybody. My name is Sofia Pereira, and I'm a senior researcher at the Faculty of Biotechnology of Catholic University in Portugal. And I have been working with beneficial soil microorganisms and also on the development of bioinoculant formulations. And these bioinoculant formulations, they are not more than the combination of these microorganisms and also some carriers that will enable their application in plants that are suffering the effects of several abiotic stresses. So this webinar will be related with this thematic, and the title of the presentation is Microbial Assisted Crop Production in Agriculture. And I'm going to show you some case studies where we apply these bioinoculants, and I will show you some of our promising results on this. So, first of all, I would like to share with you the impact or some information about the impact of abiotic stresses on crop production, and then give you some clues how to increase crop production in a sustainable way. And I will talk about the beneficial soil microorganisms, so what they are, how we can use them to help plants to tackle with the stress, and of course the case studies on the effect of bioinoculants on the improvement of plant growth. And in this case, I will focus my attention in three case studies. The first one related with saline irrigation water, the second one with the water stress in the soils, and also with the nutrient deficiency. In the case of maybe I can ask you to put the sound. Yes? Okay. So in the case of, as you know, the human population has increased in the last years and is predicted to increase even more in the next decades. And is expected to reach 10 billion of people in 2050. Yeah. And this will increase, for sure, the demand for food. And this put a great pressure on the agricultural sector, because they need to increase their production in 70%, around 70%. So this is a massive increase, and it's a goal very difficult to achieve. And associated to this demand for food, agricultural sector is facing several problems related with climate change and also with the non-sustainable farming practices. And altogether, they are creating some problems in the soil, and we have land degradation and loss of biodiversity. And when we are talking about land degradation, we are talking about an increase of erosion, a decrease in organic matter and soil fertility, and also a decrease in water holding capacity. And the loss of biodiversity will also have a negative impact on crop resilience and also in crop growth and yield. So among abiotic stresses that mostly impact agricultural production, we have those related with water. So as you know, a vast area of the world has problems with drought, so water stress. And also in the other side, we also have problems with excess of water, with floodings, which are usually related with extreme events with a lot of rainfall in a short period of time. And we also have the temperature. In some parts of the world we have heat stress, but we can also have some problems with cold. In some soils we have higher accumulation of salt, and in this case we will have salinized soils, which will be a problem to the cultures also, and also nutrient deficiency. In this case, this nutrient deficiency in soils is often compensated by the application of chemical fertilizers. And other kind of abiotic stresses are those related with the contamination of the soils. Most of times related with accumulation of metals, pesticides, hydrocarbons, among others. So it's important to say that these abiotic stresses in most cases occur simultaneously. So when we have drought scenarios, probably we also have its stress. So it's a challenge to the crops to survive and have a good growth in these conditions. So when we talk about the impact of abiotic stress on plants, we can say in a general way that they will decrease the growth and yield of plants, also the photosynthesis capacity, the lipid and the energy metabolism, and also an increase in the reactive oxygen species, which will lead to oxidative stress. In most cases, we also observed an increase in osmotic and ionic stress, and also a nutritional imbalance. It's often observed also a reduction of root length and also a reduction of water and nutrient acquisition or take. So how to deal with this problem? How to increase crop production in a sustainable way? So in the past, several strategies were applied. One of them was increasing the arable area to produce. However, we know this is a limited resource because in most of the countries in the world, it's not possible to increase the agricultural area anymore. Another strategy was the application of chemical fertilizers and pesticides in the high rates, but we know that this brings a lot of environmental and human health constraints. And another strategy, what was a good strategy and still is, it was the increasing of agricultural mechanization, but this is very expensive. So we have it's important to have other kinds of measures. So in the last years emerged a new concept related with the sustainable crop intensification, production intensification. And this aims to produce more from the same piece of land, while we are also reducing the impacts on the environment, and we are trying to preserve the natural resources and at the same time enhance the healthy ecosystem services. However, to achieve this aim, we have to minimize soil disturbance, and we can do this by enhancing or maintaining a protective organic cover, or cultivating a wider range of plant species, enhance crop nutrition, and also use integrative pest management and adopt efficient water management. And to turn this possible, we have to apply sustainable agricultural agronomic practices. And we can include here the use of improved and adapted varieties, also the application of organic soil amendments and plant cropping systems. So plant intercropping or crop rotation schemes. And of course, biotechnological tools, such as the application of bioinoculants, also called biofertilizers. And that's here that I'm going to focus the rest of this webinar, the application of bioinoculants. So we know that in the rhizosphere we have several hotspots related with the rhizosphere of the plant. So the rhizosphere is the narrow region between or around the root system, and we can observe a big interaction between the plant roots and microorganisms. And this is mostly stimulated by root exudates. And we also know that the rhizosphere is a dynamic environment where we can find a high amount of, a high abundance and also a high diversity of microorganisms. Several authors have studied the soils in the rhizosphere and non-rhizosphere soils and see the diversity and abundance of several groups of microorganisms. Here in this table, you can see just an example of the abundance of certain groups, including bacteria, fungi, in the rhizosphere and non-rhizosphere soils. And as you can see, the amount of abundance was very high in the rhizosphere, or it's higher in the rhizosphere than in non-rhizosphere soils. And that's why this is important, because we can collect some beneficial microorganisms from the rhizosphere. And here we can obtain mycorrhizae fungi from the rhizosphere and also rhizobacteria. However, it's also possible to obtain some beneficial microorganisms from the inside plant tissues. In this case, we are talking about bacteria or fungi endophytes. We can also obtain some microorganisms from specialized organs, such as the case of nodules in the leguminous plants. So in the case of rhizobacteria and bacterial endophytes, we can also call them as plant growth promoting bacteria. So these plant growth promoting bacteria, or PGPB, can have a direct effect on plants, and these effects are mostly related with mechanisms of increasing nutrient acquisition or growth promotion. And here we can highlight the solubilization of some nutrients by the PGPB, including the phosphate and also the potassium, or the production of phytohormones, such as the case of indole acetic acid, which is very important on root development, and also the production of siderophores, which are molecules able to collate hydrogen in the rhizosphere and turn it available to plants. Also, the fixation of atmospheric nitrogen or the production of ACC deaminase. This is an enzyme very important in the reducing the levels of ethylene in plants. On the other side, the indirect mechanisms are those related or can be used, this PGPB, in biocontrol strategies. And here we can find the production of antibiotics, hydrogen cyanide, lytic enzymes, and also the induction of systemic resistance. So how can we find, or how can we collect this PGPB? So we isolate PGPB from different locations and from different environmental conditions, including salinized soils, drought-affected soils, polluted soils, and so on, and also from different compartments, so rhizosphere, sediments, or plant tissues, as I told you before. And then we have a collection of this PGPB. We should test them from their ability for their plant growth promoting traits, and then we have to choose. We should choose some of them, the most promising, to be applied in the soil. So another group of soil microorganisms is the mycorrhizae fungi. So the mycorrhizae is a symbiosis between fungi and plant roots, and I can say you that 80% of vascular plants form symbiosis with fungi. And this is symbiosis, it's very important because the mycelia of the fungi will increase the area to find more water and nutrients. As you can see here in this picture, the root system, it's very developed in the mycorrhizal fungi, when mycorrhizal fungi are present in the rhizosphere. So we have several kinds of mycorrhizal fungi, but here I'm going to focus on the arbuscular mycorrhizal fungi, AMF. And these fungi form some structures inside the critical cells of the plants, including arbuscles, which are very important to change nutrients between plants and the fungi, and also vesicles, which are very important to store some nutrients. As I told you before, this is a symbiotic process where the fungi will provide water and nutrients to plants, while the plants are providing carbohydrates to the fungi. So AMF are not possible to cultivate without a host plant. So we cannot have these fungi in a petri dish, but we can have it in connection with the host plant, because they are completely dependent on them to obtain carbon and energy. And when plants have this association, they have a higher protection under adverse conditions, including water and nutrient deficiency, extreme temperatures and pH, and also problems related with pollution, such as the case of heavy metals. So once again, how to obtain this AMF. So this is a process a little bit more difficult than those related with PGPB. But to do this, we should have should proceed with the wet seeding and decantation process of the soil. Then we have to extract and isolate spores, and then we have to identify these spores. And here it's a harsh job, this part of identification. I just want to show you some diversity of spores of AMF, some beautiful pictures. Also this one, just to see the diversity that we can find. So once we have the isolation and identification done, we can proceed with their maintenance in trap cultures. So as I told you before, we cannot have this AMF in a petri dish. So we have to maintain these spores with the host in trap cultures. And after that, we can proceed with the maize seed production and to the application of this mycelia to the soil. So in summary, I can say that AMF are very important to reduce the oxidative stress in plants because they will upregulating the antioxidant systems. Also, they will increase the water and nutrient absorption to plants. They are important on the regulation of phytohormone synthesis. So all of this will lead to a higher growth or growth promotion. At the level of soil, these AMF are also very important because they will increase soil health and quality as they increase the soil moisture and also the fertility levels in the soil. However, when we think about the application of bioinoculants, we should have in mind some important issues. So the strains should be well characterized. We should know exactly which is the mechanism of action of each strain. They should be well adapted to the environmental conditions, otherwise they will die when we apply them in the soils. So they should be easy to produce and cost effective. We should know which is the best amount of cells to introduce into soils to have a good result. And also we should think about the delivery systems if we want to apply these bioinoculants in the large scale. So now I'm going to talk a little bit about the case studies. The first one is related with the application of these beneficial microorganisms on sunflower growth under saline water irrigation. So soil salinity is a huge threat for agriculture, and more than 60% of the soils in the world are affected by salt. And we know in the last decades, it was observed that the increased soil and irrigation water salinization. And we can say that more or less 40% of the total food production is derived from irrigated land. And here we have problems in more than 30% of this area is suffering the effects of salinity. So this is a very important issue for the agricultural production. And we can see here in this table some classes of soils according to the conductivity that we find in the saturation extract, and also some effects on crop yield. So the major effects of salt stress in plants are related with osmotic stress, ionic toxicity, decreased photosynthetic ability, reduced growth, water deficiency, and of course, nutritional imbalance. However, fortunately, bioinoculants may mitigate these adverse effects because they will increase the nutrient absorption by plants. Also, they will protect them from oxidative stress. They will increase water use efficiency. They will contribute to the ionic balance inside plant tissues. And of course, they will increase their growth and yield. So in this experiment in particular, we aim to assess the beneficial effects of these bioinoculants on sunflower growth in soils that were irrigated with saline water. To achieve this goal, so we swarmed some sunflower plants in pots and we irrigated them with 0, 1, and 2 grams per liter of sodium chloride, water with saline water. And after 3 days after swallowing, we introduced or inoculated our strains. So we have a control, of course, control treatment, also a treatment with PGPR, another one with the endophytes, another one with an ANF, and then a mixture of all of these microorganisms. And after 10 weeks, we determined some parameters, including soil enzymes, biomass, and ion accumulation in plant tissues. In terms of soil conductivity, as expected, of course, when we irrigated the plants, we observed after 10 weeks an increase in conductivity of the soils, as expected, of course. And in terms of soil enzymes, we analyzed the activity of catalase, acid phosphatase, and hydrogenase. And as you can see here in these graphics, the activity of these enzymes was severely affected by salinity. However, the bioinoculants increased the activity of all enzymes. In some cases, they increased activity even more to values even more higher than those found in soils without any kind of salinization. So this was a good result. In terms of plant biomass, you can see here that just after 4 weeks, we already have some differences, because the inoculated plants showed higher shoot biomass than non-inoculated ones. And this tendency was confirmed at the end of the experiment. As you can see here, we have the plant biomass in shoots and roots after 10 weeks of treatment. And we can see that the saline water decreased plant biomass, but bioinoculants mitigated these adverse effects by increasing biomass production. And this production was, this biomass production was really high in the roots. In average, we have increases with bioinoculation between 62 and 85%. So huge increases with the application of bioinoculants. In terms of ion accumulation, as expected, the increasing salinity will lead to increase in the ion sodium inside the roots and shoots. However, the application of bioinoculants decreased this accumulation, especially in the treatments with AMF and the mixture of all microorganisms. In terms of potassium, which usually compete with the sodium to go inside cells, we observed with increasing salinity a decrease in the potassium accumulation, but once again, the effects of bioinoculants increased the accumulation of this ion, particularly with the treatments AMF and the mixed schemes. So this means that bioinoculants contribute to the ionic balance of sodium and potassium inside plant cells. It's very important to avoid any kind of ionic toxicity. So, and this probably was related with the production of exopolysaccharides by the microorganisms, as they are able to bind the sodium in the soil, so preventing its absorption by plants. And also they are very, very beneficial for the structure and aggregation in soils. So in terms of conclusions, we can say that inoculation had a protective effect on sunflower and salt exposure, and AMF and mixed treatment was the treatments that better performed. Inoculated plants showed a higher biomass, higher accumulation of potassium, and a lower accumulation of sodium, which means that the bioinoculants contributed to the ionic balance of the plants. And also the application of bioinoculants improved the activity of soil enzymes, so suggesting a mitigation of salt stress in soil function. So this was the first case study that I have to show you, and then I will proceed with the next one, which is a case study related with use or the application of this PGPB on the growth and nutrient use efficiency of maize growing underwater deficit conditions. So drought has also high impacts on agriculture. According to FAO, drought is in the first place of damage and loss in agriculture by climate-related disasters, so counting 83% of these losses when compared to the other kinds of stresses that crops are suffering. So the main effects of water deficit in plants are those related with osmotic stress and oxidative stress, also damages in photosynthetic apparatus, some delay on flowering, reduced growth, and of course the decline in grain yield and quality. Once again, bioinoculants can mitigate some of these adverse effects because they are going to increase the osmolyte accumulation in plant tissues, also increase the water use efficiency, protect plants against oxidative stress, and consequently leading to a growth and a higher growth and yield. So the aim of this particular study was to evaluate the effect of PGPB inoculation on maize growth under different water regimes, 80%, 60%, and 40% of water holding capacity. And we also have a secondary aim, which was to assess the influence of inoculants, so the amount of cells that we have to inoculate to have a result. on plant growth, of course, under the same water regimes. And here, when we talk about the inoculicides, I'd like to give you some ideas about this. So I collect some information from other studies, and as you can see, the inoculicides, in this case, varied between 10 to 7 to 10 to 9. But it's important to know what happens when we use reduced or a lower inoculum when we apply it in the soils. So in these experiments, we applied two bacteria, two rhizobacteria, the Cupria Neptune and the Pseudomonas forensis, and we applied them singly and in combination, and at two different inoculicides, at 10 to 3 and 10 to 6. And after 8 weeks, we subject the plants to water stress. So we have control treatment, which was 80% of water holding capacity, and we consider that here plants are well watered. Then 60%, so it corresponds to the moderate water deficit, and 40%, which corresponds to the severe water deficit. And after 6 weeks, we analyze the plant biomass, nutrient use efficiency, and also the fluorescein diastase activity in soil. So in what concerns biomass, we can see here with these graphics that the water deficiency had a reduced impact on shoot biomass of plants grown at moderate and severe water deficit. We did not find any kind of symptoms of chlorosis in these water deficit water regimes, which means that which may explain the low impact on shoot biomass. However, we can say that bacterial inoculation increased shoot biomass of well irrigated plants and was even more pronounced in moderate water deficit conditions. However, we have some drawbacks here because the bioinoculants did not increase plant biomass under severe water deficit. So we think this could be related with the low survival of these bioinoculants when they reach the soil, and also with the competition with the natural community. Another important result was, despite we have the inoculicides has only marginal effects on biomass, we can say that the huge increase in biomass production was obtained at moderate water deficit when we applied the highest amount of the bacterial mixture. So this is an important result. So when we are talking about the nutrient use efficiency, we can say that bacterial inoculation enhanced the efficiency of use of nitrogen and phosphorus, and both indices were increased by bacterial inoculation even at severe water deficit. And the highest increases, however, were observed in plants under moderate water deficit when we inoculated a mixture of the bacteria, and those values were, in the case of the nitrogen use efficiency, were increased by 36%, and in the case of phosphate use efficiency, those increases were around 28%. In what concerns FDA activity in soils, we saw that water regimes negatively impact this enzyme in soils, and bacterial inoculation did not affect this parameter. So in terms of conclusions, we can say that bioinoculants promote shoot biomass and nitrogen and phosphorus use efficiency by maize plants. The size of inoculant had marginal influence on biometrical and nutritional parameters. However, the combination of strains seems to have more effect on maize development than the single inoculation. And you can say that PGPR can be used to reduce the effects of drought stress, especially in species with high water demand, which is the case of maize, in order to maintain its productivity with a less water supply. And the last case study that I have to show you is related with the use of phosphate civilizing bacteria to enhance maize growth in B deficient soils. So phosphorus is an essential macronutrient. It has several cellular functions. It's a component of DNA and RNA, and also it's vital for cell division. And it's required in high quantities in some stage development of plants, including the seed germination, rooting, flowering, and seed production. So when we have plants with P deficiency, we can see that their growth is stunted. We can see that the older leaves turn dark blue and stems are have a color green to purple. So the problem is most of the agricultural soils has a large reserve of phosphorus, but the available forms are in very low concentrations. So this deficiency is often compensated by the application of high rates of chemical fertilizers, in this case of phosphate fertilizers. But the plants will only use a small part of these fertilizers, and the rest of the phosphorus will be immobilized in the soil. So this brings a lot of environmental problems because we are continuously applying these fertilizers in the soil, but at the same time we also have a problem with the production of these fertilizers because the reserves of phosphorus in the world are decreasing and they are limited. At the moment, more or less 5 countries of the world hold up to 85% of the world phosphorus reserves. And we know that it's predicted that the peak of phosphorus will occur between 2030 and 2040. And the world reserves of phosphorus will last for 100 years. So we also have this problem. We should use the phosphorus, the total phosphorus that we have in the soil, because otherwise we won't have in the future other kind of solutions. So the bioinoculants, once again, could be a part of this solution because some of these bacteria, the phosphate civilizing bacteria, are able to convert the insoluble forms of phosphorus into available forms. And they can do this by the production of organic acids and also by the phosphatase enzymes, or the release of phosphatase enzymes. And in this work, we aim to evaluate the use of these bacteria as biofertilizers in the P-deficient soils. So in this case, we assembled a pot experiment where we used a soil with low levels of available forms, phosphorus available forms, and in the control treatment, we only use this soil without any kind of fertilization. Second treatment, we amended this soil with a soluble phosphate, and in the third treatment, we supplemented the soil with an uninsoluble phosphate, in this case, the tricalcium phosphate. And we applied the 3 bacteria, phosphate civilizing bacteria, and also a mixture of these bacteria. And after 90 days, we determined some parameters, including the biomass, and the accumulation of phosphorus in plant tissues, also the bioavailability of phosphorus in the soil, and the persistence of inoculated bacteria in soils by DGG. And as you can see here, in terms of biomass, we can say that bioinoculation in average, in the case of the control soils where we didn't have any kind of fertilization, we have increases in the average near the 20% when we applied bioinoculants. In soils amended with the soluble phosphate, we can see that we have increases in roots between 40 and 100%, and in shoots between 28 and 63%. And in soils supplemented with the tricalcium phosphate, we can see that the bioinoculations increases root biomass by 52 to 81%, in the case of shoots between 37 and 56%. So these were very good results in terms of plant biomass. In terms of P accumulation, we can say that the inoculation increased the accumulation of phosphorus in plant tissues, especially in those plants grown without any kind of fertilization. In the case of accumulation in the roots, we can only see results in the soils supplemented with soluble phosphate and tricalcium phosphate only with the application of the inoculation of bacteria B3. In case of P availability in soils, we can say that overall bacterial inoculation increased the concentration of phosphorus, available phosphorus in the soil. The strain B2 was the strain that showed the better performance by increasing the soluble phosphate or the available phosphate by 35% in the soils affected or amended with soluble phosphate and 48% in soils with pure calcium. And here we can see also that mixed inoculation promoted the availability of phosphorus in all soils. And the last parameter that we have tested was the persistence of these strains in the soil. So we were able to detect them in the rhizosphere of the maize after 2 days and 45 days after inoculation. However, at the end of the experiment, it was not possible to confirm with DGG that these strains were there, but probably they were, but we cannot confirm with the DGG profiles. So in terms of conclusions, I can say you that the phosphate civilizing bacteria increased the availability of phosphorus in soils, and this leads to the promotion of plant growth. Another important result it was that bacterial inoculation enhanced biomass and P accumulation in plants that grow without any kind of fertilization. That means that these PSP were able to solubilize the phosphorus that we have, the total phosphorus that we have in the soil. So this is an important result. And we can say that PSP can be used as biofertilizers in soils that are suffering the influence of P deficiency and may constitute an alternative to the phosphatic fertilizers or to reduce a little bit the application of these phosphatic fertilizers. So I have finished my presentation, some knowledge to the funding entities that finance these works. And I hope you have enjoyed this presentation, and I'm fully available for any kind of question. Thank you.
Thank you, Sofia. Very interesting.
Thank you. Very informative presentation.
Thank you.
Okay, do you have any questions?